CRISPR ALS innovation.
Did you know that 1 in 50,000 people per year are diagnosed with ALS? Although that might seem small, this equates to around 5,000 to 6,000 new diagnoses per year. ALS stands for amyotrophic lateral sclerosis, and it's a progressive disease where motor neurons die over time, and it leads to things like muscle weakness and other awful life-inhibiting symptoms. So those who have ALS have a really decreased quality of life as they age, and it comes from tough muscles, muscle pain, to slurred speech, extreme fatigue, and even difficulty chewing or swallowing. So there are many forms of the digestive system and even other parts of their body that are inhibited, and so this could actually slow the economic potential in the United States and every other place around the world because of the lack of job opportunities that they can find and just their inability to enjoy the company of life and truly value the quality of life. The FDA has approved certain drugs that don't actually cure ALS but help to treat its symptoms. Riluzole is believed to reduce damage to motor neurons, and while these medications are beneficial, they don't really treat all the symptoms of ALS diseases, and so the majority of ALS patients actually die from respiratory failure where they can't get oxygen from their lungs to enter into the blood. And so because of these factors, we wanted to help treat the root cause of ALS in order to further prevent these kinds of symptoms. And so the way we wanted to target this through our innovation is with its genes and its gene mutations.
Our objective is to create a minimally invasive approach to slowing the progression of ALS using CRISPR-Cas9 and iPSC. Our focus will be the SOD1 gene, as it accounts for about 20% of the cases in ALS. The SOD1 gene mutates within ALS and creates misfolded product buildup, and that leads to the degradation of the motor neurons. Gene therapy suppresses the accumulation of these misfolded SOD1 genes, and gene therapy can also almost completely protect this motor neuron. This treatment will aid in painful symptoms as well as aid in slowing the progression of any deadly damage and help with any mundane tasks that are necessary for living.
We expanded upon the utilization of iPSC to provide a genetic therapeutic treatment to correcting this gene mutation in the SOD1 gene. So iPSC essentially works to take the cells of any part of the human body, known as somatic cells, and reprogram it to its basic state in order for it to be manipulated by scientists through CRISPR-Cas9 and be put back into the human body.
Induced pluripotent stem cells, also known as iPSCs, are a type of stem cell that can be generated directly by the somatic cell. You start off with the somatic cells, and we will work to derive the cells to the ALS patient using skin, dental tissue, blood, and urine. By correcting common mutations associated with ALS in animal models and patient-derived induced pluripotent cells, CRISPR-Cas9 has been used to verify the effects of ALS-associated mutations and observe phenotype differences between patient-derived and gene-corrected iPSCs. The CRISPR-Cas9 system also works similar to cut and replace these DNA fragments that are harmful to ALS patients.
Cells start off in the embryo as undifferentiated, so they aren't specifically just skin cells or muscle cells. So that is our goal with iPSC. So we will be taking cells from the patient using transcription factors to reprogram the cell to its embryonic state and use CRISPR-Cas9 to cut the SOD1 gene and replace it with the correctly transcribed SOD1 gene using a guide RNA that will eventually replace this mutated gene sequence.
CRISPR has found to be the most effective and cost-useful gene editing tool.
Our primary focus for this innovation is to actually use iPSC because it provides a simplistic way and a targeted approach to how we can utilize CRISPR-Cas9 to edit that specific gene mutation. There have been multiple cases where CRISPR-Cas9 has accidentally edited different gene sequences, which has further harmed the patients. But in this way of using iPSC, we're able to provide this simplistic approach and a simplistic way in order for scientists to correct the gene mutation and further impact the lives of ALS patients.
This company is asking for $400,000 investment for 20% equity of the company. We will be the forefront of innovation. Life is pure and deserves genetic cure. Thank you.